everybody, and hello, everybody here in the audience, and also everybody online. Thank you for joining us for this exciting talk. It is for me a pleasure to introduce Maja Thiele from Odense University in Denmark. She's a star in the field of alcoholic liver disease. She's also a great clinician, educator. She will talk to us today about omics technologies for improved understanding and diagnostics in alcohol-related liver disease. At the end of the talk, please, everybody online, send your questions on the chat, and we will also get questions for the audience. Thank you, Maja, for coming here. We're looking forward for your talk.
Thank you so much, Ramon, and thank you for the invitation. I'm very pleased to be talking about this particular topics because it's something that if you had asked me 6 years ago, I would know nothing about. But I think the field of omics and myself have evolved immensely. So I will try to give you my version of omics from a kind of a clinical point of view, because I have to also disclaim that I'm not at all an omics person or an omics scientist. So the very finer details are probably lost on me. But I think it's these types of omics techniques and technologies are really ongoing as we speak, revolutionizing the field of biomarker discovery, of new drug targets, and how we manage patients in all. So I try to kind of pick up bits and pieces to show you, because again, we're discussing omics, but that's a lot of stuff. I'll come back to that. So we cannot, touch upon everything. These are my disclosures. Galaxy is a project coordinated by my boss, Alexander Krag, who's also professor of hepatology in Odense. We started this as kind of these bumblebees. We didn't know we couldn't fly, so we just tried anyway. We didn't know anything about omics, so we just teamed up with a lot of very skilled people from Denmark. Torben Hansen is a good collaborator. He's a professor in genetics, and Per Borg is a specialist in microbiome from Heidelberg, and a group from the Steno Diabetes Center in Denmark who knows metabolomics and lipidomics, and they have kind of taught us while we were going how to deal with these things. So Galaxy started in 2016 and ran for 6 years, and we are still generating the data, and I will show you some of it and other stuff that has not been published yet. We conducted 4 clinical studies in Odense. One was a cross-sectional study in 460 patients with alcohol-related liver disease, liver biopsy to stage fibrosis and inflammation, and ranging from no fibrosis at all until cirrhosis, but all compensated. So the early stages of alcohol-related liver disease. And these 460 were paired for age, gender, and BMI, with approximately 150 healthy controls. And this was very difficult because it's actually hard to find a majority of males in their 50s and 60s who are not taking any medication, who have no comorbidities, and who haven't been treated with antibiotics for the past 6 months. So I think the healthy control study was perhaps the hardest one of them all. And then we have 2 randomized studies, one in rifaximin and one in Referm, which is a prebiotic, to kind of try to modulate the gut microbiome and see what happens in these patients. And they have been extensively omics investigated, genetics, proteomics, the microbiome using metatranscriptomics and metagenomics, and then lipidomics and metabolomics. So with that in mind, that's basically what I know, because that's what I've been dealing with for the past 6 years. So I will try to provide my point of view for the advantages of introducing omics technologies in clinical practice in alcohol-related liver disease and the barriers that exist as well. And then I'll point down on like 3 different types of omics. The genetics, because that has been it's kind of the most mature omics we have. The microbiome, a lot is happening there, and I highlighted one of the key studies from previous years. And then finally, this spring, we published a large study on proteomics in this ALD cohort, and I'll dive more deeply into those data. So omics is basically all the way characterizing the host all the way from genes into gene transcriptions, into the proteins that come from this transcriptional activity, and into the modulated metabolites and lipids circulating in the body or being present in tissue. But we should also consider the environment in these omics. So the environment influences all the omics. And besides the environment and the host, we have the global microbiome, not just microbes, but also virome and fungome. Virome and fungome are, there's very little evidence on that because it's super difficult. But viruses exceed our microbiome in the millions, or even billions. So the viruses are also very, very interesting. But all these signals work obviously together. And what we have seen for the past maybe 10 years in omics studies are mostly single omics. So highlighting genetic findings or protein findings or metabolites. But what we see more and more coming together is an actual integration of multi-omics. The lowest hanging fruit is the genetics, and then looking at the transcription from genes to messenger RNA, and then again into protein targets. This has been done very nicely, was published, I think last year in Nature Genetics in NAFLD. I haven't yet seen similar studies in alcohol-related liver disease, but for sure this is coming. And moving on, so the different omics types, the genomics, transcriptomics, etc., how may we use them in clinical practice for biomarker studies? Well, you may say there's the kind of the population-based type of risk stratification, and then there's the prediction, and then finally all the way down to personalized medicine. And I think we'll see in the next decade how this opens up new avenues using these different types of omics and the integration of multi-omics. So for the population-based users of omics, obviously genetics is something we have already seen, how you may on a population basis foresee disease trajectories in different kind of risk strata of patients. But it may also be how the environment may affect risk of disease, not just in liver disease, but in general. The challenge here is that we still lack, from a liver perspective, an understanding of the different types of omics and their kind of natural state. We simply lack a reference value for a lot of the omics we're dealing with, for example, the microbiome. So there's still a lot to come on a kind of population basis, or a lot to do. Then for the precision, this is obviously a place where we can use highly effective predictive markers for choosing which patients are likely to develop adverse effects of medications and should not be given the medication, and in contrast, which patients are likely to benefit highly from treatment and should be given that treatment. And in this sense, all these types of omics seem very promising. And in other fields, there have been indicators of omics being able to, for example, predict weight loss in obese patients, weight loss according to medication, but also weight loss according to physical activity. So the challenge, of course, is to translate these omics that comes with, billions of single variables or single molecules into something that is manageable in clinical practice. And we are not at all there yet. Some of these technologies require very specialized equipment that is not, available to clinical care. I know in Denmark they are trying mass spectrometry proteomics equipment at our main hospital, but this is for a very narrow indication and for rare diseases. So we are still seeing kind of the translation from bench to bedside. And finally, the very personalized medicine, being able to kind of do a fingerprint of an individual person's risk of disease, trajectory, and response to medication. But again, here we are in a very immature stage, also because it turns out a lot of these omics vary significantly within the same patient from day to day, for example, diet and the microbiome, or even from morning to night. So there's a lot of inter-individual variation that we need to solve. Then the final, I think, very interesting possibility using omics is the possibility to try to disentangle tissue-specific effect or organ-specific disease and then general effects. We all know about people who are smoking or drinking alcohol in excess, and some of them will die from cardiovascular disease, others will die from liver disease, and another group of persons will die from cancer disease. And we are very good at risk stratifying on kind of the chance of a patient getting ill or dying, but we are really very poor at actually predicting which type of disease they will die from. So in alcohol-related liver disease, we can, with a pretty certainty, say that these patients will highly likely experience liver-related events if they progress to advanced fibrosis, but there's still about maybe 20-30% of patients who will die from other non-communicable diseases. So having omics to kind of disentangle the organ-specific risk in a certain person would be, in my opinion, very interesting. Okay, I just have so a lot of potential, and yet we have absolutely no approved omics-based biomarkers. And why is that? Well, it is difficult, and there's also a lot of barriers similar to a lot of advantages and possibilities. And one is the regulatory issues, and I'll show you some data afterwards, but costs and resources is a major obstacle as well. So now a whole genome sequencing costs around $1,000, which is extensively cheaper than just a decade ago. However, it's still a very expensive test if we were to implement it in the clinical setting. Similar with the microbiome, if you do meta-transcriptomics and metagenomics, it easily runs up to 1,000 or 800 euros. So it's very expensive tests, and they require highly specialized personnel to do the measurement and to interpret the results. Then there's the analytical validity. I'll come back to that later as well. And then there's the issue of using mainly untargeted approaches where the results come out as semi-quantitative measurements, and I will show you some example later. But for the approval process, this is a big issue. And finally, we see still in a lot of omics studies, due to this kind of untargeted semi-quantitative measurements and exploratory studies, we still see the hypothesis-free analysis primarily. And these are, again, this is more discovery and less implementation, right? So we need still to move the evidence from the majority of studies being discovery studies and until more hypothesis-driven studies. So we need to mature the evidence base. So regulatory-wise, if we want an omics-based biomarker, for example, a diagnostic biomarker in alcohol-related liver disease, we need to go through the European Medicines Agency or the FDA if it's in US, or other types of medical agencies. And as you can see here, for biomarker studies, they are very focused on a single molecule. So their approach is really not up to date with the omics studies, which generates several thousands of analytes. So you see from the EMA, it's a biological molecule, that's a biomarker in EMA standards. And for FDA, it's a defined characteristic and an indicator. It's not in the plural. So they are still very focused on one molecule. And this limits the regulatory process, and they are a bit slow on catching up on what is happening in research. So the EMA only 2 years ago published a statement on how in the future to deal with omics technologies. So they are still catching up. And then there's the length of the regulatory process. So this graph is from our collaborators in Nordic Bioscience. It's a Danish biomarker-based company exploring markers of collagen. And they listed, so they took 5 years to develop the assay to measure ProC3, which is a pro-collagen of type 3 used in liver disease. So 5 years first, and then 6 years of clinical investigations and of several publications before they even got a letter of intent, which is not an approval from the FDA, but is the first step in the approval process for the FDA. So if you want to if you have an exploratory or promising biomarker and want to actually take it into use, you need very big companies like this to push this forward. And this is something we often call the valley of death, because it's you have the academics on one side of the valley and you have the commercial part on the other side of the valley, and in between there's really nothing. So we need to bridge the academics and the biomarker commercial developers to be able to bridge this valley of death. It's not easy. So biomarker validity is something that is, important for single molecules, and it as important for omics-based biomarkers. And for clinical validity, we need to consider both the internal validity of a biomarker and the external validity. And for this, the omics field is really still too immature. So I mentioned the variance, intra and inter individual variants for many of the omics markers. But external validity is the same. So we have a lot of promising exploratory studies and then no follow-up. And I hope this will change in the end, because we need to reduce waste in research and actually focus on, when we see a good result, we need to validate it in a clinical setting. And then there's the issue of the main omics technologies using untargeted approaches, and untargeted approaches end up with semi-quantitative results. So you will not get a concentration. So this is a study that we have not yet published on Galaxy data, looking at lipidomics and changes in lipid classes in patients with alcohol-related fibrosis. And interestingly, we found that sphingomyelins were very strongly negatively correlated with fibrosis stage. So this is the fibrosis stages. So already from stage 2, there was a kind of downregulation of sphingomyelins in our cohort of 460 patients. And this trend was similar according to the level of inflammation in these asymptomatic patients. However, this has nothing to do with lipid concentrations. This is all relative to a kind of a standard within that same study. So if we were to apply the same technology on another cohort, we may get different results according to which standard we were using as reference. It's not impossible to do something, about this. So what we did was look at from the sphingomyelin class of lipids, looking down at the individual lipids, and we found that this sphingomyelin of length of 41 with one double bind were the single lipid with the strongest association to liver fibrosis stage, not just in the circulation, but also in liver. And so this lipid we could classify according to whether patients were in the upper half of the kind of semi-quantitative concentration or the lower half. And there was a clear survival benefit. So the patients with the lowest levels of sphingomyelin 41 had a far worse prognosis during follow-up, during around 4.5 years of follow-up in this cohort. However, this is again, everything is relative within that same cohort. So if we want to move on and actually try to exploit this particular sphingomyelin, we need to first develop an assay to measure the concentrations of the sphingomyelin. Then we need to validate that it's also a prognostic marker in other cohorts using concentrations. And then we need to develop, validated cutoffs, etc. going on. So it's a very lengthy process from the first untargeted semi-quantitative measurement and then into something that's actually being put into use in the clinic. So moving on a bit to the different types of omics, genetics again has been with us ever for a long time, and the Human Genome Project really revolutionized things. And I think we are on the verge of actually using the individual risk SNPs that we probably all know of. There's a handful that has been very well validated in large cohorts from across the globe. These individual risk polymorphisms. And we did a study published this year looking at the degree of fibrosis and what kind of predicted higher risk of fibrosis. And you see as the third strongest predictor of higher degrees of fibrosis was TM6SF2 and PNPLA3, which is very common, like 25%. Has at least one risk allele, was also a significantly predictor of higher fibrosis. So it's definitely promising. This study by someone here in the room also showed how we may use these individual risk alleles and combine them, not into a polygenic risk score, but just a genetic risk score. So this is just based on 3 SNPs. And obviously it's not a perfect diagnostic marker, but I think for genes which we have with us from birth, to be able to actually, label patients as having 3 times higher risk of alcohol-related cirrhosis and complications due to that from when people are born, is a very good kind of achievement for genetics. So again, on a population level, we may not use it on an individual patient because it's still too inaccurate. But on a population level, I think these genes are highly valuable. And then adding presence of diabetes, you get an even higher predictive power. But again, when you look at diabetes, it's right here and now. Again, genetics, we have it with us for life. Now, polygenic risk scores is the next step in genetic analysis. So we know these types of individual monogenic risk factors. So the BRCA gene for breast cancer, very, very strong effect sizes of a single gene. And then we know, on the other hand, the GWAS analysis, where we find rather low effect sizes for commonly occurring SNPs, like the PNPLA3. But then the polygenic risk scores are taking in the thousands of SNPs and combining them into one score. And this increases the effect. So it kind of looks like on the right, you kind of add genetic variables. You do first a GWAS analysis, and then you add and add and add until your predictive power peaks. Whenever you add more variables, the predictive power goes down because then you just introduce random variation and noise. So this is the principle of assembling a polygenic risk score. So if you meet someone who claim they are doing polygenic risk scores, but they only combine like 10 different SNPs, that's not a polygenic risk score. It's in the hundreds or sometimes even in the thousands. But when you do this, you are able to actually assemble polygenic risk scores for several commonly occurring traits, such as alcoholic cirrhosis, or obesity, or hypertension, or whatever. And this has been done I haven't seen a polygenic risk score for alcohol-related liver cirrhosis, but I have seen for audit as a measure of magnitude of drinking, and alcohol use disorder as a measurement of kind of the psychological or aspect of overuse of alcohol, the dependent and the harmful drinkers. And very interestingly, these polygenic risk scores, they do overlap, but not at all completely. So there are very significant differences between what characterizes the trait of drinking in excess versus what characterizes the trait of drinking on a kind of psychologically unhealthy manner, I would say, bordering towards dependence. So again, they did a GWAS analysis. I believe they used the UK Biobank, they used the US population, and could be an Icelandic population, so the whole of Iceland has been genotyped. And then they did this GWAS analysis for both these traits and assembled a polygenic risk score. And the first thing is to then take the polygenic risk score and look at the individual SNPs and see which cell types are these SNPs expressed in. And you can learn a lot from just doing that. So for audit C, again, this is the magnitude of drinking. Then the brain is really very highly represented in the kind of the genetic risk of drinking in excess. And you see liver. Liver is very symbolic, marked with a brown. It just crosses the FDR approved threshold for excessive drinking. And then for the dependence-wise drinking, the CNS expressed genes are still, big, but they're not as big, funny enough, as just drinking in excess, and the other genes do not cross the significance threshold. And then finally, I just want to show you this graph because I think it says a lot about drinking patterns, and we should know this when we deal with the patients in our clinic with alcohol-related liver disease. So these are then traits that also have polygenic risk scores published, and you can look at the genetic correlations between the traits of drinking in excess or having alcohol dependence and other types of traits. And there's a clear difference here. And this has to do with genetic traits for years of education, for IQ, and for age when you give birth the first. So if you give birth, if you give birth to your first child early on, you're more likely to have genetic traits to alcohol use disorder and less likely to drink in excess. Same with education. If you have a genetic genotype that indicates longer education, then you also have shared genes for drinking in excess, but not for alcohol use disorder. So again, different combinations of genes, and this can tell us something about kind of what drives the different types of drinking patterns. And then finally, what you can also use these polygenic risk scores is Mendelian randomization, which is a novel technique where you combine polygenic risk scores for one trait and look how it correlates with polygenic risk scores for another trait. So this is the same paper that we are hopefully soon getting out, published, looking at these sphingomyelins, and we found polygenic risk scores indicating higher sphingomyelins. And then we looked at how did this polygenic risk score correlate with other polygenic risk scores for alcohol-related liver disease. So with that in mind, you could see that there wasn't actually a correlation between the two. And this tells us that it seems like alcohol-related liver disease is causing lower sphingomyelins, not the other way around, not lower sphingomyelins causing the alcohol-related liver disease. So the Mendelian randomization methodology can give you an idea, at least, of causal direction. And I think this is interesting. And if you have further questions, Tommy Suvataivel did the analysis, so I will not be able to tell you any more details about this. Okay, moving a little bit on about the microbiome. So the gut-liver axis is very interesting, especially from a liver point of view, and I would say especially from an alcohol-related liver disease point of view, because alcohol enters the gut, and the first organ it meets is obviously the liver. And what we need to address when considering the gut-liver axis is all the other environmental influences on the gut. So alcohol is a strong modulator of the gut microbiome, but there are several others. Obviously, nutrition being one important. But then after entering the gut, the alcohol may induce changes to the actual taxonomy of the bacteria, so the composition, which types of bacteria are in the gut, but also the activity of the bacteria. What are they actually doing, and especially which products, which metabolites are the bacteria producing, which may then enter the portal bloodstream and eventually go into the liver. I think we will skip this. So I think one of the very interesting studies published highlighting the importance of the gut-liver axis in alcohol-related liver disease was this from 2019 from Bernd Schnabel's group, looking at cytolysine coming as a byproduct from Enterococcus faecalis. So it's not produced in the host. It is a kind of a type of cytokine only produced by the bacteria. And the first thing they did was looking at Enterococcus faecalis and the cytolysine in patients with alcoholic hepatitis, in patients with alcohol use disorder but no alcoholic hepatitis, and then healthy controls. And it seemed like it was a kind of pretty good distinction. It wasn't alcohol in itself that increased the presence of cytolysine producing Enterococcus faecalis. And the next step was very important, namely that the cytolysine positive patients with alcohol hepatitis was dying in excess. So this product from a bacterium in the gut seemed to be really detrimental for patients. And next step was introducing it into rats. So you can take stool from patients with alcoholic hepatitis, 2 patients without the cytolysine producing bacteria and 2 patients with, and then give it to mice. And this is shown here. So these are just the controls. The red and the orange are stool, the ALT levels in rodents provided, with humanized stool from patients with cytolysine producing Enterococcus faecalis, and the green and the blue ones did not have Enterococcus faecalis producing cytolysin. And you see, when you have the cytolysin present, you have far higher ALT levels, and you also have more collagen production, which I think on a longer scale, beyond if you survive the alcoholic hepatitis, this is also important. And finally, if you then provide the patients with a phage, so a virus that infects bacteria, and this phage is targeting the bad Enterococcus, then suddenly you attenuate the damage from cytolysin. So I think we are now kind of beyond the smoking gun of you can actually make interventions that targets gut bacteria, thereby improving the prognosis of patients. Okay, for my final minutes, I just wanted to give you the last omics, proteomics. I think this proteomics has the largest potential in the field of diagnostic and prognostic biomarkers. At least when we compare the predictive accuracies of our different types of omics in the ALD cohort from Odense, proteomics always comes out on top as by far the more predictive. So this year we published a paper on proteomics markers. We used the two cohorts that I told you about before, the ALD cohort with a biopsy to verify fibrosis, the healthy matched controls, and then finally we also had a validation cohort of patients with a biopsy and all had a liver stiffness above 8 because this is our new kind of protocol in Odense. So we do not no longer biopsy patients if they do not have elevated liver stiffness. So in all these patients, we had the histology, and then we did proteomics in the circulation, so in plasma, and then we also did proteomics in liver samples to be able to kind of pair the circulating proteins with the liver proteins, looking for proteins that were co-dysregulated. Now, sorry, I'll just skip that. I think this is basically in my opinion, most interesting graph from this paper because it shows 46 proteins that were co-dysregulated. So they were both correlated with fibrosis stage when looking in the liver and when looking in the circulation. So out of more than 5,000 proteins in the liver and more than 500 proteins in the circulation, 46 of them were co-dysregulated. But it was not always in the same direction. So I just want to point you to, let me just find this one. The yellow one, that's a protein related to the coagulation. So it is increasingly upregulated in liver when more and more fibrosis appears, but then it's increasingly downregulated in circulation. So this, I think, it's just a nice slide to show how we may actually use these types of untargeted omics analysis to learn something that could potentially explain pathophysiology. And if you're interested, this liver proteome website contains the summary statistics for all the more than 6,000 proteins in relation to the liver fibrosis stage and the inflammatory stage and the steatosis score. This is when compared to the healthy controls. And again, I won't go into details, but I just want to highlight that the kind of the global proteome in the healthy, it is somewhat similar to those ALD patients without fibrosis or with minimal fibrosis, but already from stage 2, something which at least I didn't think were a very advanced stage of alcohol-related liver fibrosis, we see global changes in the proteome of the patients. So clearly, there's something about the transition from no or minimal fibrosis to significant fibrosis, so stage 2, where the patients are crossing some kind of dysregulated threshold, in my opinion. Okay, we will just skip this. So what we did next was then take the proteins in circulation and assemble 3 different panels. One for diagnosis of significant fibrosis, one for diagnosis of mild hepatic inflammation and more, and one for any steatosis. And they had very, very good performances. So the 9-protein panel for fibrosis diagnosed significant fibrosis with an area under the curve of 0.9. And so other types of circulating markers for significant fibrosis have substantially lower diagnostic accuracies. And importantly, fortunately, we validated this in the independent cohort of 63 patients. And when comparing the proteomic panels with other markers of fibrosis, inflammation, and steatosis, for example, transient elastography using FibroScan for the fibrosis and CAP for the steatosis, they performed in the very top. I just want to mention briefly that often it's stated that these types of omics may risk, they are overfitting, they're using like a black box of AI where you cannot really make any meaningful algorithms for diagnosing after the analysis. And indeed, Lili Nui, my co-author, she made all types of very, very fancy bioanalytical methods for variable selection. However, for all the panels, she ended up using logistic regression because it was simple and it was the most robust. So there's no black box in this. And when we look at the 9-protein panel for fibrosis compared to the 6-protein panel for inflammation, there is overlap, but it's not big. So again, these are slightly different proteins predicting fibrosis and inflammation in the liver. And when comparing with the steatosis panel, again, very little overlap in proteins. And the final thing we did was to test how they performed as prognostic markers, looking at survival-free of liver-related events. And again, these protein panels performed very impressively good. So with that, I just want to say that I think as a physician and as a researcher in alcohol-related liver disease, I think alcohol-related liver disease, they're killing our patients at a far higher speed and a far larger magnitude than any other liver disease. So we should prioritize alcohol-related liver disease and research more on them. And I think omics is a way of doing this. And with that, I thank you very much. It's an excellent question. So from a biomarker or test point of view, I think we have the challenge of FibroScan that it's very much a monopoly and it's a very expensive equipment, and it's mainly suited to be used in liver clinics, as it is now at least, with the cost and the kind of it's a technique that you only use for one thing. Right, so with ultrasound equipment, you can use it for several things. So with that in mind, something that can be used in circulation as a biomarker would be very nice, because then you could use it in small hospitals and you could use it in primary care, etc. Another added value of these types of omics-based markers, which I haven't, touched too much on, is that, there have been published proteomic studies in cardiovascular disease, in type 2 diabetes, renal dysfunction, etc. And some of these, when we measure more than 6,000 proteins and only end up using 9 of them in a panel, another 9 proteins could perhaps be used to predict kidney disease, and 4 other proteins could be used to predict cardiovascular disease. So, at least in theory, what could happen in the future is you make one blood sample, you run it through a mass spectrometry, and then you get your risk of liver disease and your risk of heart disease, etc. This type of organ-specific risk prediction that I alluded to in one of my slides. However, we are not there at all because we do not have a mass spectrometry, equipment in hospitals. And so far, the stuff that they used to measure in our study, it was, the same size as this room. The equipment was standing in. So we need leaner machines and cheaper machines. And then we need tools that can guide the interpretation of all these omics. So we need some kind of smart software that can take all the different proteins and give you, sort of when you deliver a genetic test, right? That can give you, okay, this is the risk of liver cirrhosis, this is the risk of heart disease. And I think with how fast technology moves now, this is something that is possible within the next 10 years, for sure, but we're not at all there yet. So for this biomarker with the proteins, what we are doing now is trying to translate each protein into ELISA tests. So we are still doing it the very old-fashioned way. And I think for some time it will still be like that. It's a very, very good point. And I think you touch upon kind of the very issue of translating omics from exploratory to implementation right now, that we haven't solved this issue. I think, again, for now, I think we need this triangular regime that you are referring to, where we use some untargeted omics for discovery. And then we have a list of metabolites, lipids, proteins, whatever. And then we kind of take them into another type of assay that is quantifiable. And then we do the validation for now. But again, when I talk to some of these people, so Mathias Mann is he's one of the best proteomics people. He is convinced that we will do mass spectrometry in the future. And then the issue is no longer whether you do triangulation between, targeted and untargeted approaches or semi-quantitative or not. Then the issue will be transferable between types of equipment. And that would be the type of validation then we would need to do. But what we are doing now is the very simple method that you outlined. We're only taking those that we're interested in and then doing hypothesis-driven testing. But probably we will overlook some important biological signals by doing that. That's a good question. I think my first takeaway was with the genetics is that it potentially could alleviate at least some of the stigma related to our patients with alcohol-related liver disease, because we have some patients where the liver disease related to alcohol will be driven by excessive use, but they will have a very different profile, genetically at least, from the alcohol use disorder patients. So we could target intervention towards that kind of genetic risk score, because people who drink in excess but have no indication of dependence need other types of intervention than those who then drink in excess but have an intervention genotype. And I think with genetics, it can be a bit fatalistic, but it can also be liberating. You can say, okay, this is not because, you are lacking self-control, or your mother didn't stroke you across your head when you were small, or whatever types of stuff are being said about alcohol dependence and overuse. This is something that we actually have treatments against, and this is a risk that you carry with you from birth, just from your genes. But also, the ability to stratify the treatment or the intervention according to kind of the genotype would, I think, be an advantage if we are ever able to do this on a larger scale, obviously. Yeah, right. Good questions. First thing, the kind of the liver secreted proteins. So I didn't go into details here, but the heat maps also show which types of proteins they are, and obviously a lot of them were collagen related and fibrosis related collagens. However, it was not the majority. The majority were actually inflammation and immune system related proteins, which I think is very saying about what is driving the disease. That's one thing. And obviously, I think the immune associated proteins, they could be from the liver, but they could obviously also be from other tissues. We did an analysis on using proteomic libraries where you can label proteins as being, are they predicted to be secreted mainly from the liver, or are they predicted to be belonging to the secretome or from other tissues? And in that analysis, I believe approximately 50-50 of the main co-dysregulator proteins were predicted to be liver secreted versus 50 not. So that was about it. And for the second one, I think you're absolutely right. Some of the signals will be, beneficial tissue repair processes, and we could go into looking, specifically at those proteins, but we haven't done so yet. But for sure. Ooh, that's a good problem. A good question. I think we corrected for gender when doing the analysis, but I have to get back on that. I would expect differences for sure. I think, again, so markers of hepatocellular failure, that's maybe in your area, Ramon, and the, because this population that we have investigated in the Galaxy project is, early stage alcohol-related liver disease. So I think what we would need and what would be highly interesting is to kind of increase the spectrum and have everything from the kind of the healthy at risk, those that are drinking in excess, but their liver is, without harm, to those that have a very severe alcoholic hepatitis with clear failure. Oh, you're really finding your best questions. So I'm really happy that this was the last question, because it's again excellent. I don't think we should avoid trying to integrate the different omics. For sure not, because if we manage to actually integrate, then the opportunity to detect biological processes and gut and host interactions is magnificent, looking beyond just the individual omics. What happens now is that we see studies, doing multi-omics, but what they are really doing is, several individual omics in the same patients, but then analyzed each omic for itself. So without integrating, the closest I have seen is this study that was in NAFLD patients, going from risk polymorphism, so from a gene level and GWAS analysis, into transcription, so translating the SNPs defined by the GWAS into gene activation, and then with this gene activation, further translating into proteins, into, I think it's called PQTLs, so protein quantitative trait something. And this made it possible for the investigators, and I can send you the reference if you're interested, to detect 16, putatively causal genes in the progression from NAFLD with simple steatosis to NASH. And this is the closest I've seen. What we are working with currently in the Galaxy Consortium is different types of bioinformatics approaches, and it has proven very, very difficult because they come in very different formats, the omics, and they are analyzed, I think culturally with different types of bioinformatics methods. And if you combine it all, it's just the sheer volume of data. And then the last thing that is a very big challenge is you want the kind of biologically plausible route. So you don't want to go from proteins to genes, you want to go from genes to proteins. So you want the kind of the layers to be aligned. So your analysis should follow the biological kind of line of effects. And we are trying different approaches and are currently looking at KEGG pathways and trying to analyze KEGG pathways. But this is, I think, beyond my level of understanding. But I just know it's hard.
Well, thank you so much, Maja, for your amazing talk. Thank you for your audience. Thank you for everybody that connected through online. And I wish you all a very happy holidays. Thanks again, Maja.
Thank you so much.